Welcome back to the DrSnowBrainHealth show. I'm joined by Dr. Alan Snow. And have you ever been in a store where you look at all of the options in the aisle and they all look the same? Like everything looks the same. It's hard to distinguish between them. Well, I feel like that's true when talking about diseases of the brain. We talk about Alzheimer's, we talk about Parkinson's, we talk about CTE, and it's really hard to distinguish between the different effects of what each of these does to the brain. So, Dr. Snow, can you talk a little bit about what are the similarities as well as some of the differences between different kinds of brain disorders?
You know, it's funny because when I was looking at this, I forgot about dogs and cats. So remember for me to bring up memory loss in dogs and cats, which are different than humans and what you see in the brain. So Alzheimer's disease, we talked about brain plaques and tangles. So brain plaques, again, are these Swedish meatballs that accumulate in the brain. They look like big boulders or meatballs. They're in between neurons and they consist of a protein called beta amyloid protein. So you have that in Alzheimer's disease. You also have neurofibrillary tangles. Tangles are inside neurons. They consist of a protein called tau protein. So they also contribute to memory loss because they're causing neurons to die. That's why it's a neurodegenerative disorder, Alzheimer's disease. What about Parkinson's? And we're talking about Parkinson's is different because it's a different protein involved. We're not talking about beta amyloid protein that you see in Alzheimer's. We're not talking about tau that you see in Alzheimer's. Although this is really interesting that Parkinson's people can have Alzheimer's as well. So you get a crossover. You can have plaques and tangles and Lewy bodies in a Parkinson's patient. So Lewy bodies consists of a different protein called alpha-synuclein. And just like tangles, it's inside neurons, but it's in a different area of the brain. It's in the substantia nigra. So substantia nigra is on both sides here. And Alzheimer's is more frontal cortex, hippocampus. Parkinson's is more involved with movement disorders, and that's why they have the tremor, the rigidity, the shaking, the balance problems, and it has to be with dopamine types of neurons. With Alzheimer's, there's many different types of neurons, cholinergic type, and they used to go after acetylcholine. The original Pfizer drug Aricept was actually trying to increase acetylcholine levels, but found out that that didn't really help very much. In Parkinson's, you're trying to increase dopamine levels because the dopamine gets really lowered, and that's why you have movement problems. Now, CTE, which stands for Chronic Traumatic Encephalopathy, they have tangles in the brain, and this is after repeated blows to the head. So you see it in athletes, football players, soccer players, hockey players, skiers, anybody who has head trauma, repeated blows to the head. They get tangles in the brain, but it's in a different area again. It's in the grooves of the actual brain called the sulci, and we can show a picture of what that looks like. So some of the similarities are like you have tangles in Alzheimer's, you have tangles in CTE, but the differences are it's in different areas of the brain. So that becomes very important on what you see clinically. And then you also have, interesting, because you talked about dogs and cats can get memory loss just like humans, and it's called doggy dementia or canine cognitive dysfunction or feline cognitive dysfunction in dogs or cats, usually over the age of 7 years old. But guess what? They have huge plaques, but hardly any tangles. So they're having memory loss, these dogs and cats, because they're having huge amounts of giant boulder plaques that is, again, reducing the connections to remember thoughts. That's why they have memory decline, cognitive decline. And there's a bunch of stuff that occurs in other misfolded protein disorders. I've studied amyloid diseases for about 30 years. And these are misfolded proteins, much what you see in the brain, except it's in the body. So you could have stuff accumulating in your kidney, in your heart, in your liver, in your spleen, in your GI tract. Guess what? They're different amyloid proteins. So they're different proteins. They're all folding into a similar manner. And my theory has always been that heparan sulfate, actually, these proteoglycans, are found in all these diseases. I actually wrote a paper, I gotta look back, maybe late 1980s, where I showed that the common constituent in all these different protein misfolding disorders was the accumulation of heparan sulfate proteoglycans. And we found actually heparan sulfate accumulates before the proteins are misfolding, so it actually may be the cause of many of these misfolded protein disorders. And we're going to talk about that when we go about the real cause of Alzheimer's. I think it's heparan sulfate proteoglycans. I've done a lot of research on it. Showed that it's in the amyloid plaques and cerebrovascular amyloid deposits in Alzheimer's brain. Then I also showed it was in Down syndrome. And guess what? In Down syndrome, it comes into the neuron like one day after birth. It was just loaded. And it's also in the Lewy bodies and Parkinson's. I'm looking to see and doing some work with Anne McKee out of Boston University to see if the heparan sulfate is accumulating in the tangles in CTE. And I've already shown that it's accumulating in all these other types of misfolded protein disorders.
So I want to talk a little bit about the symptoms. You've done a great job of teasing out some of these key aspects, but you talked about how there's tangles in Alzheimer's and there's tangles in CTE, and they're in different parts of the brain. I think that is a really interesting observation, but I'm also interested, what kinds of effects do we see of those tangles in the different parts of the brain in which they accumulate?
Well, tangles in Alzheimer's is probably contributing to neuronal loss. If you have neuronal loss, you're going to have memory deficits. So, Alzheimer's, the main thing is memory. And it gets so bad that, these people don't even know where they are, where they live, who's their loved one when it gets really bad. And they don't recognize. I remember my mother had Alzheimer's, and I clinically just dealing with it. She knew who I was for the first few years, but it got worse and worse. She didn't even know who I was. And then they lose their speech and all that kind of problems. In CTE, think about football players, it's in a different portion of the brain, and they get emotional outbursts. So they get some different clinical signs. So it's in different areas of the brain that's not necessarily involved in thought and memory. It's more involved in emotion. And so in the grooves of the brain is very involved in emotion. And then in Parkinson's, when you're in the substantia nigra, that area, that's all about dopamine and movement. So they're losing dopamine levels, the dopaminergic neurons are being decreased, and then you start to see a lot of the balancing problems, the tremor in Parkinson's. You know a Parkinson's patient right away. As soon as you see somebody standing there and shaking and can't control their shaking and what's called tremor and rigidity and other types of balance problems. In dogs, it's interesting because they have plaques in the brain, not very many tangles, but they're still getting memory loss. So what's their cognitive decline? My older dog used to go into a corner and they'd stare for 10 minutes, just stand there, look in the corner, right against a wall. I think I'll show some pictures where my editor actually found a cat that went into a corner and would stare at the wall, and definitely had feline cognitive dysfunction. Yeah. So it's definitely, you're seeing different clinical signs with these different diseases. Now, I was talking to you about some people with Alzheimer's will have Lewy bodies in the brain, so they have Alzheimer's and sort of Parkinson's. So sometimes there's mixtures as well, as you see in these brain disorders. It's very hard to find CTE and they have Alzheimer's or Parkinson's, because these are mostly younger athletes.
What about dementia? Where does dementia fit into this whole equation?
So, it's interesting because the word dementia, they used to bring out a lot of Alzheimer's patients will get dementia, but you can get dementia by having a brain tumor in your hippocampus, okay? So when somebody has Alzheimer's, they're trying to rule out what other things you can have that could show the same cognitive decline or memory loss. If you have a tumor sitting in your hippocampus, you probably have symptoms like Alzheimer's, so they got to rule it out. They'll do a brain scan or a PET scan, MRI, also look for tumors, throughout your body and make sure that you don't have cancer that's, somewhere in the brain. People have glioblastomas also. It's a tumor in the brain. It can show some signs of cognitive decline and memory loss as well. So it's a mixture, and you got to really go after, like, what could he have, go after the diagnosis. Remember, we talked in Parkinson's, if you take the cerebral spinal fluid, now you could do this seed amplification assay where they take alpha-synuclein and put it into the CSF and it starts aggregating. I can actually diagnose that you have Parkinson's or will get Parkinson's even before you have the clinical signs of it, which is amazing.
So we talked a little bit about different parts of the brain. Do we see that each of these diseases tends to attack different parts of the brain, or is there overlap? And what do we see as being some of the differences when, for those who are not as familiar with the different parts of the brain, where you find plaques, tangles, you see this inflammation, you see these Lewy bodies, like what parts of the brain do these types of misfolded protein disorders, where do they start to appear?
So it's a good, the answer is there's a lot of overlap and it's hard to distinguish, because what happens in Alzheimer's, it spreads from one area of the brain to another. So you could have it in your hippocampus, then it goes to the cortex, and it's in the cerebellum. You can see plaques everywhere. And there's no quite defined limit. Just like in Parkinson's, where you have individual treatments because it's varied from individual to individual. Same thing happens in a lot of these neurodegenerative diseases. You can't put it like, oh, it's always going to be in this area of the brain. What they find in Alzheimer's is that it tends to spread from one area to another, and it gets worse and worse. And then, prion diseases are proteinaceous infectious particles. Actually, we should talk about that as well. So it's a different protein discovered by Dr. Stanley Prusiner, won the Nobel Prize in 1997. I actually worked with him, showed that heparan sulfate proteoglycans were in the plaques in prion disease. Some really rare disorders, Creutzfeldt-Jakob disease, Gertzmann-Straussler syndrome, Kuru. Kuru is where the cannibals used to eat the brains of their loved ones before they put them in the funeral home, and then what they found is that by doing so they got the same disease of a prion disease and they died. So that's why people were stopping cannibalism, because you can actually spread. Prion disease is a spreading disease, so it's an infection. It's like having a virus basically. But instead of a virus, it's actually a proteinaceous, a protein, or it's a protein that spreads. And so when Dr. Prusiner came out with this theory, everybody thought he was nuts, and they said that's impossible for a protein to be spreading through the brain, and he turned out to be right, and that's why he won the Nobel Prize on that. Think about mad cow disease as well.
Well, this has been very helpful, Dr. Snow, talking about some of the different diseases, where they show up in the brain and how they affect our brain health. So I look forward to many more conversations about how to understand these disorders and how best to preserve your brain health over time.
Yep. Okay, Scott, thanks a lot.